Hello. First of all, I'd like to thank the Oxford University Biological Society for inviting me to give this talk. I was due to give it in April earlier this year, but because of COVID, that wasn't possible. Nevertheless, I'm really looking forward to sharing our research results with you and also to hearing the questions you may have at the end. I'm going to be talking about a general problem in biology, which is how the remarkable variety of tissue shapes and forms that we see around us are generated.
Each of these shapes, the daffodil, the brain, the leaf, the butterfly, the pitcher leaf, and the heart, all arose through sheets of cells somehow miraculously deforming themselves into these different forms. And the question I want to ask is, how did that happen? And might there be some common fundamental rules by which such transformations occur? Now, the idea that there might be unifying principles or rules behind a whole series of different transformations isn't new. A few years ago, I attended a conference in which the poster for this conference showed a series of creatures or forms generated through origami. Now, origami involves a series of basic transformations or basic folds, and it's the combination and the way you make these folds that generates all these different forms. But the fundamental principles are actually very simple, just by folding a piece of paper. The question is, might there be an equivalent set of rules or principles that underlie all the varied forms that we see in biology? Now, unlike origami, however, in biology, we don't have an external hand that's doing all the folding, that's doing all the deformations or transformations that we're seeing here. It's all occurring through the actions of genes within tissues. It's internally driven through the properties and behaviors of the cells in a tissue. And so the question is, what might be the principles through which the equivalent of origami occurs in biology? And to illustrate that, what I'm going to do is trying to do a kind of biological origami, the equivalent of origami, and see if we can achieve it, but through biological properties, internally driven, rather than through externally imposed hands performing the operations.
So our starting point for this bio-origami, as I'm going to call it, is going to be a sphere of tissue. You can think of it as a sphere of cells collected together. And this tissue, the first problem we're going to have is to generate our sheet, because origami starts with a sheet, but in bio-origami, we're going to have to somehow generate that sheet to begin with. So how might we do that? Well, to generate a sheet, we need some sort of orientational information that's going to allow us to sort of grow or deform this sphere in one direction more than another. And to do that, I'm going to introduce the notion of what I'm going to call a midplane identity, an equatorial region that runs through our entire sphere that is going to help us establish this orientation system. Now, just to show you, this region in green, which I've called the midplane identity, runs through the entire sphere. I'm going to cut a section through it so you can see it runs all the way through. And you can think of this as a region of gene activity that is somehow defining an equatorial plane or midplane. Now, in order for such a thing to help us orient growth, we're going to need to have, as I said, some sort of orientational information. And so to do that, imagine there's something propagating out from this midplane. It's producing something that, say, diffuses away from the midplane, and that we're able to take the gradient of that substance. So if you can take the gradient of this substance or propagating material that comes out of the midplane, we'll get a set of arrows. So the arrows tell you the steepest gradient of propagation. All right, so the steepest gradient of decline in the concentration of this substance. Now, once we have a set of arrows like this, which we call a polarity field, we can define two types of growth. One type of growth is we can call parallel to the polarity field, Kper. So that's growth oriented parallel to the local polarity. So for each region of tissue which has an arrow, we can say the tissue might grow at a certain rate parallel to the orientation of that arrow. And we can also define a second growth, which we can call perpendicular to the polarity. So it's in the plane perpendicular to that polarity. So growth can also be oriented like that. And so we can play with these two different growth rates for each region of our tissue. We can say to what extent does it grow parallel or perpendicular to the polarity. So in my first illustration, what I'm going to say is suppose now growth is only perpendicular to the polarity. So that in each region, growth is going to be high, perpendicular to the polarity, and little or absent in terms of parallel to the polarity. So Kper is uniformly going to be high throughout our tissue. So if we grow our tissue in that way, what will happen to our sphere? Okay, so if we do that, what happens is the sphere flattens. It grows into a flat sheet. And just to complete the tissue, we end up with a disc. So our sphere has grown into a flattened disc by invoking this polarity it created through this midplane. Okay, now we've created a disc, a sheet. What if we want to bend the sheet, as in origami? So how might we do that? Well, here's an idea. Suppose that we have a strip of higher growth rate on the upper surface of our sheet. All right, so high growth perpendicular to the polarity on the upper surface of our sheet. Might that cause a bend? And the reasoning being that if we have more growth along this line and the top compared to the bottom of surface, that might somehow create a bend. So is that what happens? Well, if we run this, what you generate is a sort of bend, but it's not an origami fold. All right, we're not getting a nice, neat origami fold. We're getting a sort of curved fold. And so why is this? Why aren't we generating a nice, neat origami fold? And what would we need to do to the system in order to enable us to generate a nice, neat origami fold? Well, to answer these questions, I'm going to now give you a demonstration. Okay, so here I've got a piece of paper. It's a normal piece of paper from a printer. And the only strange thing about it is that I put tape on one side of the paper. And there are 3 strips of tape, one going along the top, one going down the side, and one diagonal strip. And what I'm going to do is just cut the top strip off to begin with. Okay, so I'm just going to do that with these scissors. So here is the top strip. Now I'm going to take some water over here, and I'm going to put this strip into the water, and I'd like you to think about what's going to happen. Okay, so try and predict what's going to happen. So I put the strip into the water. Okay, let's see what happens. You can see it's starting to curl to form a ring. Okay, so why does it do that? The reason is that the paper, when it absorbs the water, wants to expand, okay, get bigger. But the tape doesn't want to expand. It wants to stay the same size, as it were. And so there's kind of a conflict between the paper growing larger and the tape remaining the same size. And by bending, the conflict is resolved because it allows the paper to get long while it allows the tape to stay the same size. Okay. So what's going to happen now when I take the vertical strip, the other strip? Will it do the same or something different? Okay, so let's cut that off now. All right, cut the ends off. I put it in the water. What do you think is going to happen? Take it out. Okay. So what's happening? It's curling again, but now it's curling in the other orientation. It's forming a long tube instead of curling on itself in the longitudinal direction. So why is that? What that tells you is that the paper is anisotropic. It preferentially expands in one orientation compared to the other, And that's why we're getting these two different results. The fibers actually in the paper are aligned in a particular way so that you end up with the paper stretching in different orientations. So now we've done that, let's take the final demonstration, the diagonal strip. And I'm hoping that you can predict what's going to happen. So now let's take that. I'll cut this off. Try and think what's going to happen now when we put the diagonal strip. In the water. Okay. Now I don't know how many of you anticipated this, but we're now getting a nice helix. Okay. So what this tells you is that the paper is anisotropic, and by having anisotropy or preferential orientation of fibers in the paper and preferential orientation of strength in the paper, we're able to orient the direction in which this folds on itself. Okay. So armed with that knowledge, we can now go back to our example of why the disc folded in a particular way. I hope you can appreciate now that the reason that it's folding in this strange way is that the growth in the upper surface in this line is now going in both directions. It's going both parallel to the line, creating this arch, and perpendicular to this orange line, creating the fold. And the result is we end up with it arching or bending in both directions. So if we want to get around this problem, what we need to do is somehow orient the growth in the plane of our tissue, in the plane of our disc. And one way of doing that is to create a second polarity field. So here now I'm showing you two polarity fields. One that you've seen already with the black arrows, what I call the orthoplanar field, that emanates from this midplane. The other one almost goes orthogonally or perpendicular to the blue arrow, to the black arrows, these blue arrows, and they go from one end of the equator to the other end. All right, so to creating an alternative, what we'll call a planar polarity field. And the reason that this is called a planar polarity field will become evident when we now try and grow our disc. So let's grow the disc just as we did before, orienting the growth with this orthoplanar field such that growth is perpendicular to it. All right, if we do that, but now show what happens to the planar polarity field, what we see as we grow it is that the blue arrows now get deformed and start to align in the plane of the tissue. All right, so I can show you that in a completed disc. So now we have a field of arrows, a planar polarity in the plane of our sheet. And now when we want to make our fold, we can specify three types of growth direction. There's what we call Kper, growth parallel to the black arrows. Kper2, growth rate perpendicular to this but parallel to the blue arrows. And a third growth rate, Kper, which is perpendicular to both. And if we have it such that the growth now in our blue line, is only going to be, which is on the top surface, only happens parallel to the blue arrows, parallel Kper2, in other words, it's only parallel to these blue arrows, what's going to happen? Let's have a look. It folds, it gives you a nice origami fold. So, to summarize, we can perform a kind of bio-origami, but to do so we need a number of ingredients. First, we need a set of regional identities, like that green midplane region, equatorial region. Then we need two orthogonal polarity fields. These are the blue and the black arrows, two sets of arrows that are going to allow us to orient our growth according to three different directions, as we've seen with this cross shown here. And finally, we need mechanics. We need the tissue to be mechanically connected together so that it deforms in a coherent whole. And with these principles, we can do bio-origami. We can create these types of folds, but now using the internal rules of growth of the tissue. But we can also do a lot more. you may notice that with origami, we can only create a rather set of angular flat shapes. It's rather limited in what it can do. But it turns out with these rules, we can do much more than origami. We can create rounded forms, forms that are much more typical of the things that you see in nature. So far, I've given you some illustrations of the principles by which tissues might be produced and deformed. But the question is this true? Is this the way that actually tissues do behave? And to evaluate that, we need to look at specific biological cases and see the extent to which what I've been telling you helps us understand what is going on. And the first example I'm going to give concerns how a flat leaf develops. This is a leaf of the workhorse of plant genetics, Arabidopsis thaliana. And we know quite a lot about the genes controlling the formation of this sheet of cells that forms the leaf. And to give you an idea of where these genes are active in the leaf, what I'm going to show you is a kind of a small section through the leaf, looking in cross-section to see where these genes are switched on. And when you do that, what you see, this is shown on the left here, what you see is that we essentially find three domains of gene activity, three regions that are distinct. There's the light blue area, the top half of the leaf, which is called the Ad-abaxial domain. Then there's a sort of brownish coloured area, which is the Ab-axial domain. And then finally, I've colour-coded the midplane region in green that runs between the two. Okay. Now, these genes don't just simply decorate or identify different domains of the leaf. They also are important in the formation of this sheet-like structure. Because if you were to mutate these genes, inactivate one of them, let's say, you end up with a leaf such as the one shown here, which is now radially symmetrical. It's a radially symmetrical mutant because we've lost the blue domain, we've lost the midplane. All we're left with is the abaxial domain, the brown domain, and the result is a radially symmetrical mutant. So to illustrate that, I'm going to show you it in a different species, Antirrhinum or snapdragon. So here is the normal snapdragon leaf, and here is one of these radially symmetrical mutants where these domains have been inactivated. So now I'm going to play that movie. So you can see that this radially symmetrical mutant really has lost its sheet formation, its inability to form a sheet. So how do we explain that in terms of the principles that I've gave you earlier? So let's go back to our sphere with its different polarities. If you remember the blue and the black arrows, and this third one, the orange one, which is perpendicular to both. And let's grow it again, as shown here. And remember that the reason it forms a flat structure is because growth is being oriented by the black arrows. That's what's keeping it. So growth is being oriented from the plane perpendicular to the black arrows, which is giving you this sheet-like structure. It's no longer a disc. It's narrowed at one end more to be like a leaf, because in addition to growth being oriented by the black arrows, we're also reducing growth parallel to the orange arrows at one end of our structure, which is what gives you this narrowing at one end. All right, so but using this, we can generate a leaf-like shape. And if we look what happens to the blue arrows, we again see they're in the plane of our sheet. Now, what would happen if we were to take away the midplane? Use exactly the same model, but take away the midplane and lose those black arrows. Okay, well now we've lost the black arrows on the right, and we've now only got the blue arrows to orient our growth. All right. And if we now, so we've lost, in other words, our ability to make a sheet. So if we now run this model exactly the same as before, but without the black arrows, we end up with a cylindrical tube. So we can explain the formation of our radially symmetrical mutant as a mutation that essentially gets rid of the black arrows of the orthoplanar polarity field. And so we only end up with one polarity field, and that's why we end up with this cylindrical form rather than the sheet that we see normally. All right, so we can understand these mutants and how they act in terms of growth being oriented by these polarity fields that I've been describing. That tells you something about how the sheet is formed, but what about the shaping of the sheet? Why does the sheet get shaped to produce a leaf with its characteristic outline? To understand that, we need to go and think more about how growth is oriented in the plane of the sheet, okay, after the sheet has formed. And we can do that by imaging the development of a leaf by fluorescently marking its cells. So here, for example, is a leaf bud of Arabidopsis, labeled with its cell outlines labeled with a fluorescent marker. And if we grow that leaf, as it grows, we can track and follow all the cells, all the vertices that connect the cells, and extract all the growth parameters involved in the formation and shaping of that leaf. Once you have that, you can then start to come up with models for how the leaf gets shaped and how the cells get divided as the leaf is shaped. And that's illustrated here. So here's a model now in which when we run this model, we'll see that the leaf is growing, the cells are dividing according to certain rules, and the pattern, the shape and the pattern of cells reflects very much what you saw in that movie. All right, so we're now capturing much of the behavior that we see experimentally, but using a growth model based on the principles that I've been telling you about. And the heart of this model is a polarity field, a planar polarity field. That's what's orienting the growth. It's these arrows, which are black in this case, but they correspond to the blue planar arrows that I was showing you earlier, that are orienting the growth, that allow, for example, this narrowing to occur at the base of the leaf, because growth perpendicular to these arrows is reduced at this end. And that's why you get a PTO. So this shaping of the leaf is really guided or depends upon this polarity field running through it that's helping orient the growth and allows you to shape it in this way. Now, we proposed this model about 8 years ago, but people said, look, it is all very fine, but there is no evidence for any of these arrows. Where is the biological evidence for these arrows? And in fact, they were right. If you look at a leaf, and here's an Arabidopsis leaf again, if you look at the cells, this is now in a mature leaf on one side, you'll see that there's no obvious polarity, there's no arrow, there's no difference between one end of this leaf cell to the other end of this leaf cell. Okay, or this cell, all right? They all seem to be like rather symmetrical jigsaw pieces that look the same from top to bottom. But then a few years ago, we introduced another fluorescently labeled gene called basal. And when we introduced this gene, which was fluorescently tagged into the leaf, what we saw was that it only labeled one end of the cell. So if you look at this cell, for example, the signal, this orange signal, not orange, it's sort of greenish signal, is only at this bottom end of the cell. It's not at the top end of the cell. This one is the bottom end of this cell. And so you can see, using this marker now, we can assign a set of arrows. Every cell actually does have an arrow. It has one end that's different from the other end. It has a polarity. And moreover, you can get the average polarity across the tissue. You go over the entire leaf and you quantify this polarity. And what you find is you end up with a field of arrows. And this field of arrows is very similar to what we've predicted by the model. So we now have direct evidence for these arrows, this polarity, exactly as we might have predicted, further sort of confirming the validity of these types of models. Okay, so what I've shown you then is using the principles that I've outlined, we can explain how something like a flat leaf of Arabidopsis, how the sheet is formed, and how the sheet is then shaped to produce a leaf with a particular outline. So we've seen how the principles I've described can account for the development of something like a flat leaf. But not all leaves are flat. So can the same principles be used to explain more complex leaves, let's say have cup shapes, or the leaves that we find in carnivorous plants, which can be very ornate indeed? So to address this question, we started a project several years ago on studying how carnivorous trap leaves are generated. Here you can see 4 cup-shaped leaves in carnivorous plants. They've all evolved to trap animals within these cups. Right? And what's remarkable is that this has happened 4 times independently. These have arisen 4 times quite separately from flat-leaved ancestors. All right, so the question is, how are these cup-shaped forms that we see here, how did they evolve? And how is it that it happened so many times independently? And we've been investigating that problem by studying this species, Utricularia, or Bladderwort, which is convenient because the cups are much smaller than the other species. They're only a few millimeters long, and they can be imaged quite readily under the microscope, allowing us to see exactly how this cup-shaped form arises. So just to show you what this Utricularia looks like in 3D, here's a 3D movie of the Bladderwort that we've taken. You can see it looks quite ominous. It's only, remember, a few millimeters big, so it's not quite as scary as it looks. But if you're a small crustacean and you're unfortunate enough to swim by and trigger one of these hairs, then you'll get sucked inside and get digested, unfortunately. So how does something like this develop? Well, we can look at the cells at an early stage. You see it starts from this small bud and it forms like a cup shape. This is in section. It forms a cup shape, then almost folds over to almost a spherical form. And then finally it enlarges like this. And this little gap here is the mouth that will form the mouth into which the prey gets sucked. So how is it, first of all, that a cup-shaped sheet arises? And that seems to be quite different from the flat sheet that we've seen in species like Arabidopsis. Well, we can turn to the genes involved in sheet formation because we know quite a lot of them, as I said, in Arabidopsis. So just to remind you, we have these genes in the upper surface or the upper region called the Ad-abaxial genes, and the ones in the lower surface called the Ab-axial genes, and then the midplane between the two. So one of the questions we asked was, where are these genes active in something like Utricularia that forms a trap? Where are they active in the primordia that form the trap? And so we tried to localize where they were expressed using various probes, identified the corresponding genes from Utricularia, and the results are shown here. So the signal, this for example is looking at the blue gene, the Ad-axial genes on what's in the upper surface, upper region of the Arabidopsis leaf, and the signal is shown as this darker color. Okay, and I hope you can see that the blue gene, as it were, is expressed preferentially on the inside of our trap, on the inner surface, whereas the brown gene, the Ab-axial genes that are normally on the bottom of the flat leaf, are expressed on the outside layers of our trap. Okay, so we can summarize that by this arrangement. So here's a flat leaf where you have the blue on the upper, brown on the lower, whereas in Utricularia, in a trap, we have brown on the outside and blue on the inside. So the blue inside corresponds to the upper surface of the flat leaf. And the question is, how did this arrangement arise based on the principles that I've been describing? So let's go back to our sphere and remember that to generate a flat leaf, we had a polarity field, these black arrows, orthoplanar polarity field emanating from this green midplane. And that was enough to generate a flat sheet. Supposing we were to displace this midplane so that instead of running through this along the equator, it displaces northwards at its edges. So we formed a kind of curved region here, a curved green interface with the ad-axial, the blue domain, being in a smaller part compared to the brown lower domain. Well, if we run exactly the same model as before, but now with this displaced midplane, what shape will we get? So let's find out. So this is the model. We're now running it with a smaller blue domain than we had before. If you run that, you generate a cup shape. If I just show you this with a sphere, you'll see the entire cup. So actually, it turns out to be quite straightforward to generate a cup. All you need to do is to have a smaller ad-axial domain, a smaller blue domain displaced, and you will generate a cup very simply. And that makes a very strong prediction. It predicts that the blue domain in the Utricularia primordia should be smaller, displaced and smaller, compared to the blue domain or ad-axial domain in the Arabidopsis leaf. And this is the data. So here's an Arabidopsis leaf. I hope you can see that the brown signal, the stronger signal, the boundary runs pretty much through the leaf, through the middle of the leaf, whereas in Utricularia it occupies a smaller domain, all right, which is consistent with with the model that I've just given you. So it turns out that it's very straightforward now to generate a cup-shaped leaf from a flat leaf. All you need to do is to displace this ad-axial domain, this blue domain. And maybe that's why it's happened so many times independently. It's actually not very difficult to generate a cup. You just need to make this shift in gene expression. And you can only understand that once you understand how these polarity fields are controlling the growth. And in fact, it turns the situation on its head, because it's harder to make a flat leaf than it is to make a cup. Because for a flat leaf, you need to make sure the plane goes through the middle, whereas for a cup, all you need is to be displaced at one end. So actually, in some ways, from a developmental point of view, cups are easier to make than flat sheets. So we've seen how the cup-shaped leaves of carnivorous plants can be generated by the principles that I've been talking about. But these leaves are not simply cups. They have themselves quite complicated shapes. And you can have, of course, cups in many different forms. So how does that shaping happen? And again, we're going to go back to the Utricularia to look at its later stages of development, to try and understand the principles by which the more complicated shape of the cup is formed. As you may recall, after forming a cup, the early Utricularia bud transforms itself into this kind of almost spherical shape, which we're seeing in section. And then from this sphere, there is a further deformation and growth, which leads to the mature trap, all right, with its characteristic mouth for trapping animals. And during this process, some regions grow more than others. For example, this magenta region, which is in the bottom, just below the mouth, like the chin of the trap, grows quite straight and long to form this elongated edge, faster than this red region around it. So you end up with an alteration in shape because some regions are growing faster than others. The question is, how does that happen? And how can we explain what's going on? So let's start off with a simple model to try and get our head around what's happening. In this model, we have a sphere. We're going to approximate the early structure as a sphere, and we're going to divide it into domains, like the magenta domain and the red domain, and see if we can, just by playing with these domains and influencing the growth rates within these domains, we can generate the transformation. Now, although this is a sphere, unlike the spheres that I was showing you earlier, this is not solid. All right, it's got a hole in the center because we're dealing with this cup-shaped form that has arisen, that's turned, as it were, into a sphere. So it's hollow in the center. And we can also decorate the sphere with cells. We can put cells over the sphere so that we can monitor how these cells also get deformed and change shape as our tissue grows. So the first attempt at modeling, we tried to just see if we could explain the shape transformation by playing with how fast different regions grow relative to each other. And what you're going to see here is our sphere growing with different growth rates, particularly higher growth in that magenta region that will form the chin. And I've color-coded the cells according to the area of the cells. Okay, so let's see what happens if you try and simulate the growth that way. So you see the tissue grows, the cells get bigger, but instead of generating a nice straight chin, what we see this bulge forming. All right, we're not forming this nice elongated straight edge. And the reason that bulge is forming is because if you increase the growth rate in a particular region like this, and growth is equal in all directions, it's isotropic, then the tissue will bulge out. All right, it's just going to happen automatically. So how is there any way around that? Well, again, the trick is to play with polarity. So imagine now we have a polarity field in our tissue, just like we had a polarity field in our Arabidopsis sheet. But now the polarity field is over a sphere, over a curved surface. It's the blue arrows, the planar polarity. And let's suppose it's running from the stalk towards the mouth. And now we can have very similar rules before, enhance growth, for example, in this magenta region, but have the additional rule that growth is going to be higher parallel to the blue arrows than perpendicular to the blue arrows. So that's going to orient our growth. Okay, if we do that, then we can have a much straighter edge. We're starting to generate a shape that's much more like the trap. So just by playing with polarity and having this anisotropic growth, we can now generate a much more realistic shape. So is there any evidence that this is correct? Well, one of the predictions of this model is that it predicts that the cells are much more elongated in this chin or ventral domain. And I've color-coded here the cells according to the degree of anisotropy. So the more elongated they are, the yellower they are. So you see, we predict a set of yellow cells in this region, highly anisotropic, but we can actually measure the anisotropy of the actual cells in Utricularia by imaging them. And this is the data now. This is viewing it viewing the ventral side. This is viewing it from the side. And you can see that indeed the cells are elongated in this region, in particular, the region we predicted based on this model. So that's gratifying. We're seeing a correlation or a corroboration of our predictions based on cell shape. But what about the polarity? We're postulating there are these hidden arrows, these orientations within the tissue. Is there any evidence that that's the case? Well, in Utricularia, if you look in the inside of the trap, what you'll see are these hairs all lining the inside of the trap, the internal cavity. And if you zoom in on those hairs as shown here, you'll see that two of the arms are more splayed out than the other two arms of each of these hairs. And that allows you to assign an arrow to each hair, a polarity. The hairs have a polarity. That's a bit easier to see on the next slide. Here we've got a 3D image of one of these hairs. And you can see that the two of the arms are more splayed out. And then we can place points on these hairs and automatically then extract polarity, an arrow from each of these hairs. And if we then do that for the entire trap, all right, we go through and we place these points and define these arrows on all the internal hairs in the trap, we can then get their field of polarities, the set of orientations as a whole. And if you do that, then what you find is that the arrows have a coordinated pattern, and that pattern tends to be that they point from the base of the flower, from the stalk towards the mouth, which is exactly what we predicted based on our model. All right. So that's another prediction of the model that's borne out by the experimental data. So what I've tried to show you today is that armed with these concepts of regional identities, polarity fields, and mechanics, we can explain not only origami forms, but a whole range of biological forms. And moreover, it's allowing us to test these models and make predictions that we can validate with experimental data. So we're starting to build up for the first time a coherent logic and experimentally based set of models that allows to explain the range of different forms that are generated. And of course, that raises a whole set of new questions. What controls these polarities? How are these regional identities connected with these polarities? How are these polarities influencing the growth and the deformation of cells? These are all exciting questions that lie ahead. And so that eventually maybe we'll have a fully mechanistic understanding of how these remarkable and beautiful structures are produced. Everything I've told you today was the result of a team effort. People coming together from different disciplines and working collaboratively to produce the results that I've shown you. So before finishing, I'd like to just thank the people involved. First of all, I'd like to thank Richard Kennaway, who's seen here in between programs having a bit of light relief. He's a brilliant computer scientist, and without his contribution, all the modeling that I've been showing you about growth really wouldn't have been possible. So I'm enormously indebted to Richard. I'd also like to thank many other members of our group. Here we all are having tea together. This is in the pre-COVID days. There's Beatrice Pinto-Gonçalves, Chris Whitewoods and Karen Lee, who really did the work on the Utricularia project and developed that whole set of results that I showed you. Kathy Mansfield, who did the work on quantifying the polarity of the basal protein in Arabidopsis, and Sam Fox, who did the work on quantifying the cell divisions and the growth, tracking the growth of the Arabidopsis leaf. I'd also like to thank Geordie Chan, shown here hugging a fellow person in the lab, who was the first really to demonstrate that basal showed this coordinated polarity in the Arabidopsis leaf. And many other past members of the group who contributed in various ways to this project, particularly Jay Cheng, who did the modeling of the 3D cup that I described. Finally, I'd like to thank two collaborators, both computer scientists, Andrew Bangham and Przemysław Pruszynkewicz, and both of them have been inspirational in the discussions and the ideas that we've bandied about. They've been fantastic set of collaborators, and many of the ideas that I presented really arose through discussions with them. So I'm eternally grateful for that. So on that note, I'd like to finish and thank you very much for listening, and I look forward to all your questions.